So again, viruses are obligate intracellular parasites, and they're using your genes. So let's just take a cell. This is a normal cell. The virus, and I always draw them as little diamond shapes just because I've gotten the habit of doing that, will inject its genetic material into the cell somehow. Okay, there are different techniques for how they do it. Frequently, that DNA will go into the nucleus and it'll insert itself somewhere into the nuclear genome. And from there, it's replicated into messenger RNA that comes out and binds to this rough endoplasmic reticulum where different genes are turned on. So now what it's going to do at this point is it's going to start enslaving the cell. It's basically taking over what's going on, and it's having the cell make more copies of itself. There's genetic material that goes into the cell. So it's making copies of the external envelope. Now from there, depending on the type of viruses, you can basically separate them into two groups. You have naked and enveloped. A naked virus, that's it. They have the nuclear material, whether it's DNA or RNA, and they have a capsid coat. That would be it. Now, some of these will egress out of the cell, so they will have a bunch of these new viral particles line up and bunch up at the surface, and they'll bleb out. So they'll bleb out, and they'll be surrounded with the membrane of the cell they just infected. So if you had hepatitis, you told me not to give you another disease today, so bless your heart, you had hepatitis. Well, the hepatitis viruses, they're different kinds. There's RNA, there's DNA, there's single-stranded, there's double-stranded, all that cause hepatitis. That's just the location in the body where they're causing the disease. But a lot of those will infect, and then they bleb out, and they've got some of the skin, the cellular membrane of the liver on the outside of it. That gives them some, have you ever heard the term tissue trophism? Okay, tissue is aggregation of cells that are organized to do a particular thing. Okay, trophism is eating. So a hepatitis virus tends to attack the liver. Okay, where you've got other viruses that will attack blood cells. Ebola viruses attack connective tissues. So they can have very specific tissues that they like to infect. Some viruses like to infect the lungs. Okay, those can be really bad too. All right.
What inside the cell is actually replicating the virus?
All of your machinery is going to be included in that. So if it comes in, it inserts its DNA, then it's going to be producing messenger RNA. And many times the viral messenger RNA will have a higher affinity for your ribosomes than you do.
So the cell's ribosomes is making virus?
Preferentially, yes. So it treats it just like it does its own DNA, except in many cases the viral genomes or the viral genes are turned on constitutively. They're always on.
So the virus's genes, it has to insert into the cell's genome in order for it to be reproduced, or can it?
Some can be free-floating for a while.
And then what reproduces it if it's free-floating?
Well, they'll have to carry their own enzymes if they do that. Viruses are so diverse that there's almost no one set rule for them. And when it comes to the naming system for them, the taxonomy for viruses, it confuses the heck out of me. The reason is because they've changed the naming nomenclature over the years. They started naming it after people. So there's some viruses that have a name that sounds like Smithii or something like that. Well, then they changed that and they started naming it after the tissue that it infects. Hepatitis viruses. Even though the hepatitis viruses, there's 7 or 8 of them, and all of them, they're not genetically related. They're different kinds of viruses. they started naming them after the geographic location. Ebola Zaire was first isolated from people on the Zaire River. So God only knows how they come up with the names for these things. We're just going to cover some of the basic concepts of virology and a few of the diseases that are caused by viruses. You can have, I've said this before, you can have RNA-based viruses, you can have DNA-based viruses, and they can be either single-stranded or double-stranded. RNA is less stable than DNA. Single-stranded is less stable than double-stranded. By stable, I mean mutation rates. If you have a single-stranded RNA virus, that sucker's mutating like you wouldn't believe. Every time you infect a person with a single-stranded RNA virus, it may be producing hundreds of different strains from that. You get different types produced. So, he gets infected with a cold virus. 10 or 15 days later, the viruses in his body may look totally different than the ones that originally infected. Most of the strains that are produced are going to be harmless because the mutations that occurred in them were kind of detrimental to the virus. But a few of the strains are going to be better able to infect the next person over. Does that make sense? So if you were to look at viral strains, this is type A. When you start having some mutations, this is type B. This is type C. This is type D. Over time, you're going to get a shift in the antigenic determinants on the surface of these viruses. And we'll talk about antigenic shift versus antigenic drift in a little while. There's another problem you have is that animal viruses, unlike bacteriophages, you can have multiples infecting at the same time. So influenza vaccines, you're not supposed to get the influenza vaccine if you're running a fever. Why is that?
You have another virus.
You're getting another virus, but you're also in your what?
Doesn't it kill it? It won't be effective if you're running a fever?
Well, it won't necessarily be effective. It could cause some immunity later on, but here's the danger. You've got this cell being arranged and this viral particle being arranged. And here's the chromosomes for them. Let's say that there's one chromosome each. Some of them have multiple fragments, some of them have circular, some of them, they're all different. But what can happen is you can get these things mixed up. So A, B, C, D, E, F. These things can recombine inside the cell, so it gives you A, B, F. So now the vaccine would have worked here, but this is the wild type. So now we have a wild type that's being produced that has characteristics of the vaccine, and the vaccine is no longer functional against it. So you'll get all sorts of weird mutations when you mix the vaccine with someone who's got the disease. You never want to do that. So if the patient's running a fever, you don't give them a shot. Well, they have all these free, you go to the health department down there, usually every year they'll set up a, have you seen the drive-through flu shots? Those are kind of cool. You go in and you fill out the form at station 1, and you drive to the second one, they shoot you up, and you drive over here and you wait 30 minutes to make sure you don't have an anaphylactic reaction to the shot. You always have to wait 30 minutes after you give somebody a shot of some sort, because if they go into anaphylaxis, they can flop around like a dead fish, and it'd be bad. Used to be a lot of pediatricians would tell you what they would do. They'd give your kids their regular shots, and they'd say, The doctor wants to see you before you go. And then nurse sticks her head in every few minutes to say, he got tied up. They're lying to you. They're just checking to make sure the kid doesn't have an allergic reaction. So after about 30 minutes, the doctor comes in. Well, everything looks okay. You can go now. And the mom or dad sitting there going, why the hell did you make me sit here for 30 minutes?
Now,
Yeah,
tell the truth.
it just, yeah. When you go to a pediatrician, my criteria for the pediatrician, one, they've got to be intelligent. I talk to them and ask them a few questions. Two, they need to have a sick room and a healthy room. So they need to have a waiting area for sick patients, and they need to have a waiting area for healthy visit patients. That's a big deal to me. Because I don't want to sit there with this kid snotting over there right next to my healthy kid who's just getting the vaccine. So having those rooms separate really makes a difference. All right. So viruses can be extremely diverse in the way that they infect. Double-stranded DNA viruses are extremely stable. Guess what? Most of the successful vaccines we have are against double-stranded DNA viruses. You look at the smallpox virus. Smallpox, any of your pox viruses are big old honking blocks. They're bricks. They're huge. They fall out of the air really quickly. They're usually transferred person to person or animal to animal, and they have a huge chunk of double-stranded DNA inside their nucleus.
Now, when you say they fall out of the air, that means like they're airborne a little bit, or they don't like...
Well, if I sneezed or coughed or I'd released some of these things into the air somehow, they'd float for just, a little while and then they go down. So your chances of getting it from an airborne contaminant is less. You just about have to have physical, you can, but you need kind of physical contact, or you're in the sick room with a kid who's got the chicken pox and, you're kind of in a closed area and then you get hit like that.
And then, so does that mean the single-stranded ones like stay up there in the air longer?
Some do. A single-stranded naked virus is usually less stable, so they don't last long. But a double-stranded naked virus can float on the air, especially if, depending on the way their proteins are arranged, they can float for a while. In fact, HIV could be made to go airborne with one mutation.
So, like, what is measles?
Measles is extremely airborne. Now, let me think.
The double, single, naked?
I'd have to look to see. I can't remember off the top of my head.
So the single strands are less stable, but they replicate faster.
You have to take it case by case.
What do you mean, when you say less stable, like, what do you mean?
Mutation rates are higher.
Oh, so they change more.
They change more.
So when you say less stable, they have an affinity to change more.
Right. They have a higher mutation rate than the double-stranded DNA viruses. So, in fact, that's where I was going with this. A double-stranded DNA pox virus, chicken, smallpox, or canary. Just throwing some out there. So chicken, smallpox, or canary viruses, they have large segments of their genes that are homologous in all three of those. They don't change much. They don't mutate much, even though they're in different organisms. So Edward Jenner, who was hanging out with the milkmaids, I think I told this story before. Edward Jenner came up with his first true vaccine. Milkmaids were known to be the prettiest ladies in the area. They were basically sold into servitude. Some poor farmer had too many kids, and some of them were girls, so you become a milkmaid. You go to the dairy farmer and you get up and you milk cows every day. Well, they would get cowpox on their hands from touching the teats of the cows. And they'd get some tiny little bumps, but then their skin was smooth. Everybody else, all the rich people, had these pustules from getting smallpox. It was 80% of the population or more had those their scars for parties. Try to cover those up. So what happens is if I take, here, I forgot to put cow. If I take cow pox and I inject you with cow pox, it doesn't have the ability to make you as sick as smallpox does. But it's got enough homology, enough similarity to the smallpox that it gives you immunity to that, simply because it's not changing much. Where if you do that with single-stranded RNA viruses, influenzas, things like that, you're not going to be able to come up with one single vaccine that will work against all of them. They're working on some really cool ones. They've got one that's called a composite vaccine. Imagine, you know those little styrofoam balls that you put the fake flower arrangements in? My mom always thought she could do those pretty arrangements, and they were hideous. And oh yeah, and they'd sit on the kitchen, on the dining room table from Thanksgiving till the next Thanksgiving, and they were just ugly. Well, imagine you take a styrofoam ball and you put a protein, and each one of these proteins is from a different virus. All over this little styrofoam ball. So now I inject the styrofoam ball, and your immune system starts attacking all of those different things. So you're teaching your immune system to attack multiple different antigens at one time. A composite vaccine is composed of multiple different antigens from different places. So they're much more effective than using other simpler vaccines. Now, vaccines work great, and you have two choices with viruses. You have killed virus, or you have modified live virus. And there's pros and cons to both of those. Can you help me figure out what those pros and cons would be?
Between one and one?
Pros and cons of having a killed versus a modified live virus.
Less of a reaction of the kill virus?
Less of a reaction of the kill virus? Right. Modified live, basically you expose them to some chemical that weakens the virus. Sometimes you just heat the virus up, and that's enough to weaken it so that it's still alive, but it's not strong enough to cause the infection. That's the idea behind it. And you're right, lower reaction. A dead virus doesn't try to infect a host cell. It doesn't release all of the proteins that would be released if it does attempt to enter the host cells. So the modified live virus, even though it's weakened and it's not strong enough to cause disease, it does activate genes.
Is there more contradictions with the modified live that affect other pieces in your body? So you kind of need constant watch over one more time?
You worry about modified live viruses being able to revert and cause the disease. If you were to inject a modified live virus into someone who's immunocompromised or weak from some other disease that you don't know about, then the virus could get a foothold because the person's not got a strong enough immune system to keep it knocked down.
So the kill virus isn't going to activate genes, or is it just not going to activate?
It's not going to activate any genes. All you're relying on with a killed virus, let's say that you've got a killed virus, it's got these capsid proteins, which we'll talk about in more detail here in a few minutes. If it's naked, if it's enveloped, it'll have spike proteins. These are those N's and A's. H's and N's, excuse me. So enveloped viruses have to stick something in the outer surface so they know where to go. But if I kill it, that's it. All you're going to get is exposure of those proteins to the immune system.
So that it's training the immune system to go after those proteins.
After those proteins. But that limits the number of antibodies you can produce against that. Where if you have an activated genome, it's going to unsheath itself. It's going to try to start making proteins, and it's going to secrete those proteins. So you'll get, this is all outside the cell. If a virus inserts itself into the cell and produces some strange proteins, then you get cytotoxic T cells. These are also known as killer Ts. That will attack and kill that host cell. This would be all antibody. The antibodies would be raised against antigens that are exposed on the outside of this, where you get both antibodies and cell-mediated immunity. I know I'm giving you a lot of words here, so I'm going to sit down and talk about this for a minute. Cell-mediated immunity is where I've trained a whole army of these cytotoxic T cells to recognize an infected cell. We've got these killer Ts, and they go around and they're looking at the cells and they're trying to find out if that cell's infected with a virus. Well, if that virus is inside the cell, bits and pieces of it get expressed to the surface, and the cytotoxic T cell decides it's time to destroy it.
So when we're talking about, is there like a vaccine that's killed and a vaccine that's modified live?
Yes. The danger in the modified live is it can revert back and actually give you the disease. A lot of veterinarian vaccines will do that. And there's some of the chicken vaccines that 1 in 10,000 of the chickens that get the vaccine will actually get the disease and die. Well, they're chickens. You give the vaccines when they're chicks, so you're going to have some die off. But with humans, that's a little too high a risk for us to make a vaccine for humans to do. Now,
So the killed is better for the envelope ones, but or the modified is better for the unenveloped, or is there? Okay.
you get more immunity from the modified live than you do from the killed.
Does it have a preference for a double-stranded or single-stranded? What about RNA?
No preference.
When they say synthetic vaccines, they're talking about killed or that whole other vaccine?
Synthetic vaccine is one that doesn't contain all of the antigens. So you pull out specific proteins from the virus and do it like the composite vaccine that I talked about. That would technically be a synthetic vaccine. It attacks the cells that contain modified life. It doesn't recognize the floating virus that's floating around.
Now, when you talk about the lower reaction time and the activate genes, are those pros or cons, or does it just depend?
It depends. Okay, I know it sounds confusing, but here I'll clear it up for you really quickly. Polio vaccines. There were Saban and Salk were two researchers that were trying to come up with vaccines against polio. Polio is the reason we have a March of Dimes. It was affecting kids all over the place, and mothers got together. Mothers got together and said, we're going to do something about this. So they started collecting pennies and dimes and sending it to researchers that researched polio. Well, Saban and Salk came up with their vaccines. One of them came up with a modified live, and one of them came up with a killed virus vaccine. The modified live could be put on a sugar cube. They would take these sugar cubes and they would put a drop of the modified live virus on it and responses to make sure that 100% of the viruses were killed, that could give you the disease. So what they've started doing is they give 2 rounds of the killed virus, give you immunity to some of the antigens, and then they come back and hit you with a modified live virus vaccine. Many of the vaccines where you get multiples of them, you'll be getting killed vaccine for the first couple of rounds, and then the 3rd round will be a modified live.
So is that what they do, like when you have to have this series of shots, like the 3 series? Does that generally give you 2 doses of the killed and then they give you the live, or does it depend on?
It depends on the vaccine, and I'm not up on which ones do what. I'm thinking some of the hepatitis vaccines you can get working in healthcare.
Yeah, they 3 killed. They're all the same.
They're all killed.
Do you know the last one? They're all identical.
Well, I know the polio, they still recommend 2 killed and 1 live.
So now with polio, did I get the polio vaccine?
I doubt it. I don't think they still give the polio
They're
vaccine.
bringing it back, I know, in the military.
Or, well, the military gets everything.
Marcel's coming back, MMR's being forced back, and polio's being forced back.
MMR should be forced
Yes,
back.
because of what's happening in California, is that the waffle one?
That's smallpox. Smallpox. Yeah, because I had that one. It's the air gun, you know. And the thing is because it's got modified live virus in it, if you take that scab or you touch that pus that's coming out of it and touch someone else, you can give them a copy of the vaccine. My cousin has 2 scars because as a little boy I took the scab off of mine and stuck it on her arm. So she got a scar where I scarred her with my scab. Come on, I was like in elementary school. I was a typical boy. Just make her
Okay, so
gross out.
with the killed and the modified live, it just depends on the virus.
Yep.
Which one's better.
Yep. So what would I do if I wanted to know? I would call a virologist and study it. I would look up the information on the particular one. There are pros and cons to having both. I don't think I would want modified live unless the chance of it reverting back to the wild type was 1 in a million or more. They have a vaccine for feline leukemia virus. That's cat AIDS. Why couldn't they come up with a vaccine for human HIV? They could using that same technology.
But yeah, I don't know.
There's a percentage of them that get the disease from the vaccine, and the percentage is too high to take a chance on making a human version of it. So they're looking for other ways of giving vaccines. Like this whole cell-mediated thing, that's where the cytotoxic T cell is the cell that mediates immunity. Have you heard of DNA viruses? I'm sorry, DNA vaccines? They're kind of cool. So you take HIV and you pull out a chunk of the DNA. It's not the whole virus, so it can't give you the disease. It's just a piece of it. Okay. And you put that into plasmids. Here, I'll draw it up here. So I make a bunch of plasmids that all contain a gene from HIV. And now I want to get them into a human. So I add gold. It's an abrasive that doesn't react in the body too much. You know, you're not going to overdose somebody with it. And then they use one of those air guns like we were talking about before, and they put it into the leg. They literally blow those plasmids into the cells.
When you say they add gold, like,
There's tiny little specks of actual gold.
and why do you have to add gold?
Think of it as a shotgun blast.
Oh, okay.
Okay, this is how it works. So the shotgun blast punches holes in the membranes of the cells.
So the gold punches holes in the membranes of the cells, allowing for the
Plasmids to be taken up. So now, if I take a shotgun and I shot it right here, okay, it's going to kill the one that's directly in front of it. But some of these other cells that are behind it and over to the side, it's going to carry the DNA in there, but it's not going to cause so much damage that they die. Okay. So what happens, instead of virus here,
Does the gold stay in the cell then, or will it
Yeah.
work its way out?
yeah, it stays in the cell. I mean, it's molecular size gold. It's not like large nuggets or anything. So what happens is the plasmid goes inside the host cells. You get transcription and translation. Some of those proteins get expressed on the surface of the cell. Cytotoxic T cell comes along and it reads that and it goes, wait a minute, you're not supposed to have that antigen on the surface. That's not self. So if you're infected with HIV, you need to die. So now instead of having one cell that recognizes that, we start making a whole army that will kill any cells infected that express that antigen. So we get a different, they got the idea for this cell-mediated thing for HIV from the prostitutes in Thailand. There were a whole group of prostitutes in Thailand that were repeatedly visited by men that were HIV positive, and they never got the disease. So they started trying to research them. Actually, a lot of them quit doing the prostitution because they could make enough money giving blood. They paid them for being guinea pigs that they could quit the other life. So what happened is they had their own cell-mediated response. So they knew it was possible for cell-mediated response to give you immunity to HIV. There are another group of prostitutes from Africa, but they didn't have cell-mediated response. And they're still trying to study them to figure out why they are resistant to HIV when, they should have died from the disease long ago.
So is it possible they got hooked on getting small amounts of fluid injected into their system to where they slowly built up?
I don't know.
So those plasmids in the gold, that's part of cell-mediated response.
Well, that's a part of a DNA vaccine. I know today's a little confusing. I'm just kind of, we'll
How
go
did
back
they know
over
the
this.
prostitutes didn't get HIV?
Well, they test them for HIV, and you can test for the presence of the actual viral particles in
Well, how did
blood.
they know if they were resistant to it?
Well, 10 years of doing your job, exposed to it over and over when hundreds of other prostitutes have died.
Can they look at like their cells or whatever and see like stuff on the antigen on the outside and then
Yeah, there's whole groups of people that try to study that. And those were the two groups of prostitutes that actually worked. So they think the ones in Africa actually have developed an antibody that works pretty well at preventing it, where it's a cell-mediated response from the ones from Thailand. And it's genetics. All right. I'm going to hit you real quick with one other concept of, a couple other concepts of viruses. One is the concept of oncogenes. Do you know what an oncogene is? Cancer-causing gene? Have I said that in here before? Or did you just hear that somewhere else? Oh, yeah, that's one of his favorite things. Oncogenes. They're cancer-causing genes. The way I look at oncogenes is they're kind of like a light switch. You have some light switches that turn things on, some light switches that turn things off. So if I go up here and I turn on the light switch and lock it into place, the lights will never go off. Well, let's say that in a gene you have certain stop points that stop genes from being replicated. If I go in and I block the stop codon, it never stops. So if I mess with either the stop or the start signal for a protein to be made in the cell, then I can turn those genes on forever. Those are oncogenes. They're usually ones that control cell growth. Because, cancer is just growing out like crazy. It's growing out of control. Many viruses, because they insert themselves into your genome, will insert near an oncogene. And they cause those genes to grow out of control. Why would a virus want to do that? They're replicating themselves, exactly. So if I insert insert myself into some cells and cause them to grow like crazy, then I'm replicating parts of myself. That gets down to a concept in bacteria.
That's not how the cancer is born, is it?
It's one way.
A virus can do it.
A virus can do it. Normal mutation rates can do it. Every time you replicate a cell, you take a chance on getting a mutation form. So, in my opinion, anything that can stimulate more cell division or excess cell division can result in cancer. So you get exposed to too much sun, the sunlight is actually not only causing mutations, timers forming, but it's causing cells to die from excess exposure, and then you've got to replace them. So every time you replace them, you take a chance on getting one that's got the mutation that causes cancer. So natural mutation can work. You can have chemicals. Well, I don't know, you know there's a lot of mutagenic chemicals out there now that if you're exposed to large amounts of it over time, it will cause mutations. What about irritants? I know you've seen the mesothelioma commercials where you're exposed to asbestos. Asbestos is just, it's insulation. If you look at it under the microscope, it looks like it's got millions of points. It looks like something that, it's like fiberglass under a microscope almost. So it's insulation. So people would inhale it, and it would cause the cells in the lungs to be irritated and have to be replaced as they're dead from the mechanical action there. And that would result in a special kind of lung cancer. Mesothelioma. Anything that stimulates long-term inflammatory responses can result in cancer. Dr. Romano, who used to be the department head here, had esophageal cancer. Well, he had acid reflux since he was, in his early 20s. He always complained of indigestion, so bad. The acid reflux went up into the esophagus. It killed the cells lining the esophagus, and it caused new cells to form. So it sped up the chance of him getting an esophageal cancer, and those are nasty cancers.
Why smokers are more prone to,
It's not just
inhalants, it's the
yeah, but there's mutagens in the chemicals. There's the irritation. There's the heat. You see some people that, like Dr. Sauterer, I think he could light a whole pack at one time and go, and God, I love him to death. He's had leukemia, and I've tried my best to get him to stop smoking. I picked on him, tell him, see somebody smoking, I'm like, yeah, put another nail in that coffin. It's hard. Quitting smoking is the hardest thing I ever did. I smoked. Of course, when I was growing up as a teenager, it was cool to smoke. They had the Camel commercials. All right. Lytic versus lysogenic. It's another concept of virology. Let's take bacteria, for example. A lytic cycle, you've got a bacterium. The virus will land on the surface. It will inject its genetic material into the cell. It starts taking over, and it starts making all the little bitty parts, and it makes bunches of copies of them, but it doesn't let the cell do anything for itself. So it would be like me taking all of you to my home lot and saying, build me a house, but you can't eat, you can't drink, you can't do anything but carry bricks and, hammer. Eventually you're going to die. Well, the virus doesn't care. It wants you to die. As the cell lyses, it releases those viral particles. That's a lytic cycle. It kills immediately. I'll say quickly. Kills quickly. So it goes in, it takes over, it works them to make as many copies of itself as it can, And some of these can make hundreds, if not thousands, of copies, depending on the species. Then it releases those extra pieces. So I just made a note to myself is all I did there. Well, the problem with this routine is now these new viruses have to go out and find another host. They got to go hunt. They got to find the perfect host. They got to infect it. That's kind of a hard life. Lysogenic viruses got smart. They insert their genetic material into the genome, and then that cell goes through cell division, and it treats the viral genes just like its own genes. So with every generation, it's replicating the viral genome again and again and again. Why should I take the chance on going out and not finding the right host when they're doing the replication for me? So this goes on, yes ma'am?
So can that virus, even though it's replicating with the cell, can it leave the cell and then go infect someone else?
Yes ma'am, it sure can. Basically what it does is in stress. Things that cause stress to these bacteria would be starvation, pH stress, temperature stress, UV light, antibiotics. All of those things can cause stress to the bacteria. And if the stress is at the right time, the virus goes, oh, wait a minute, my host is dying. I need to take my chance on going outside. So, yes ma'am.
It's not stress, but let the virus get over the bacteria.
So if there's no stress, it's just going to stay.
It's going to stay until stress happens.
Does it go unnoticed? Like, does the bacteria realize, like, we know we're sick from the virus, and then
Say there was a discussion question on your last exam that had something to do with this. What was that? CRISPR system. That's a bacterial way of recognizing viruses and cutting them out. So some bacteria can detect the presence of these viruses, but it's a constant biological warfare going on between the virus and the bacteria. The bacteria that has the CRISPR system can recognize certain sequences. So that means that if the virus mutates those sequences enough so it's no longer recognized, the CRISPR system will then have to mutate to recognize the new sequences. So it's a constant biochemical warfare going on between the two groups.
what's the difference between an infected bacteria and a non-infected bacteria? They still function the same way?
If the bacteria had feelings, would it feel sick?
Well, no, it's a really good question. And in some cases, there'd be no difference in the bacterium. The bacterium grows at the same rate. It does the same thing. But there's another concept. That's the little note that I wrote over there for myself. Lysogenic conversion is where the virus inserts itself into the genome and it alters the bacteria. Lysogenic conversion. In fact, I'll write it on the board. Lysogenic conversion is kind of cool. Vibrio cholera, L. strain. the benefit, don't answer this, you answered before. What is the benefit to the virus to have the phage be more, to have the bacterium be more pathogenic?
More hosts, like the bacteria would have more hosts.
The bacteria would replicate faster, and each bacterium is another copy of the genome. So I've replicated by making other people sick. Isn't that kind of cool?
What was the question?
What's the benefit to the virus if the bacterium becomes pathogenic? And there's several examples of lysogenic conversion in your book.
So it can alter in different ways, but that's not, like, it can alter as simple as speeding up the replication.
Sometimes it gives
Or
it a
self-addition.
toxin, and they don't know where the toxin can come from the phage, or the toxin could already be in the host cell. It's just that it's not going to be toxic unless the phage inserts in there.
What do you mean the bacteria are more pathogenic? Means the bacteria is going to go infect other things?
Okay, if you get a non-pathogenic strain of E. coli in your gut, not E. coli, of Vibrio cholera in your gut, and you can get that from eating raw oysters, don't do it. But you eat a raw oyster and it's got non-pathogenic strains of Vibrio cholera, it may replicate and get 5 or 6 copies of itself before it comes out the other end. But if you get a pathogenic strain of L. tor, you got 5 or 6 million billion. I'm not sure how many.
So it replicates faster?
It replicates much faster because it's making you
So
sick.
it's making it pathogenic?
And it makes it more pathogenic. Yeah.
So the degree of something being pathogenic is how fast it replicates and what it hurts.
Right. When we get into that chapter, you'll see that there's a whole lot of people out there that come up with some fancy jargon and terminology to talk about pathogenicity and virulence factors, and they've got all of their own language. To make it as simple as possible, if it can make you sick, it's virulent, and most of your virulent organisms are going to be able to replicate at a high rate.
I learned that lipid A or whatever, you kill the bacteria and got the A something, endotoxin. The endotoxin was left over and then caused you to shock or whatnot. If all the bacteria is killed and all those viruses are released at once, could that cause you to get a viral infection? Like,
It's not a viral infection because this virus only infects bacteria. But it could release too much of the lipid and cause an endotoxic shock event occur. So I see what you're saying. So you come in and you've got an infection and I hit you with an antibiotic. Well, what happens is that antibiotic can be a stressor and it causes all of those bacteria to move from the lysogenic into the lytic phase. And it causes massive release of lipid A, the endotoxin, which causes the patient to go into shock and die.
So a virus can go from lytic to lytic.
They can go from lysogenic to lytic, but they can't go from lytic to lysogenic. If they're killing quickly, they're going to kill quickly, but they can go in the dormant phase and then decide to kill. Lysogenic conversion is where the phage, in its lysogenic form, increases the pathogenicity or virulence of its host bacteria.
so what's the point?
Repeat that? Okay. What's that? Lysogenic conversion is where the lysogenic phage increases the virulence or / pathogenicity of its host bacteria. There's several groups out there that are not pathogenic unless they have a lysogenic phage inside of them. You can take L. tor strain of cholera, cure it of the phage. They afraid it hit somebody in the eye or something.
Because I think about a virus wanting to, kill or make things sick. But I don't.
We're anthropomorphizing the virus itself. Okay, how do I say this? It's an excellent question. It's just how do I answer it? If you have a new virus that's just jumped species, there's no kind of working relationship between you and the virus. So if it jumped from a pig to a human, it's usually much more virulent for a number of years. After a while, the immune system gets pretty good at knocking the virus out, or the virus gets pretty good at hiding from the immune system, and we're all infected with viruses right now that aren't making us sick. They're present, they're there, they're just hanging out inside our cells. We've got them beat down, or they are hiding, one of the two. It's hard to decide.
Is that what it means to be a carrier?
You can be, yeah.
It's not expressing.
It's not, well, a carrier is going to be shedding the virus. You can have someone that goes into a kind of a dormancy phase where you have long periods of time without shedding the virus. But a carrier would be someone who's showing no symptoms of the disease, but they're constantly, Typhoid Mary with salmonella, the particular salmonella she had a bacteria phase, so it's lysogenic conversion. Typhoid Mary was shedding that particular salmonella out of her gallbladder all the time, but she didn't show any symptoms of being sick. It was just the people who ate her food or her cooking died. That was around the turn of the 1890s, early 1900s, and she killed a bunch of people, and they just had started one of the first health departments in the country in New York. They were doing things like trying to control sewage. You can no longer pour raw sewage down in the streets. Okay, they were trying to control things like that. So they went and they found her, and they said a lot of people have been getting sick and even dying after eating your cooking. We think you're a carrier. And she didn't believe that those little critters could make people sick. So they said you can't cook anymore. You need to find another job. So she quit that restaurant, went around the corner on the same block and got a job at another restaurant. And a few weeks later, they're like, more people are getting sick and dying. It's just from a different place. So epidemiology was coming into play then. They were learning to track the source of the infection. They tracked her down. She refused to quit cooking, so they sent her to prison. And in prison, guess where they put her? In the kitchens. Okay, a bunch of prisoners got sick. They finally released her with the understanding that she wouldn't cook again. And she tried. She tried to go sell shoes or something for a while. And it didn't work out. She was starving to death. Evidently, she was not a very good clothing salesperson. She didn't have the personality to deal with the public. You saw drawings of her from the newspapers back then. I wouldn't want her serving my food, much less selling me shoes. Anyway, they finally forced surgery on the woman to remove her gallbladder, and told her she could never cook again. And I think that's the end of the story, and all that gets back to these viruses. All right. I'm going to stop just the general talk about viruses and actually start on the lecture. So far that's just a brief overview of viruses. Now we'll rely on the chapters. Do I need to turn? How much time do I have? Oh, I got plenty of time. Ooh, it's hot back there. As the heat rises, it's a lot hotter back there. If you get too hot, you can move closer. I'll try not to spit too much. All right, so viruses viruses in virology, there's some general characteristics, general properties of viruses, nature of the virion, the virus host, and then quantification of viruses. We're not going to get into much on the quantification of viruses. Viral load is something you hear about with certain diseases like HIV, and they can determine that using Western blots or they'll use Western blots to determine the specific viral loads in many cases. Some of the triple drug treatments are really kind of cool. They're trying to hit the virus on different ways. Think about a virus. A virus has got to find the host, it's got to infect the host, and what's inside the host, it's got to replicate and then break out of the host. So each one of those is kind of a step where we can attack the virus. If we can prevent the virus from finding the host cell, we stop the viral infection. If we can stop it from entering the host cell, we can stop the viral infection. If we can stop it from replicating inside the host, we stop the viral infection. If we can stop it from egressing out of the host cell, we stop the viral infection. So many of the drugs are going to be targeting different points on the viral life cycle because it's less likely that you will get multiple mutations that allow the virus to respond, to keep going. If I just lock the door, eventually it's going to pick the lock and come in. But if I lock the door and start shooting it, then it's not going to be able to mutate against the bullets and the door. Not sure that's a good analogy, but there we go. All right, so viruses are genetic elements that cannot replicate independently. They are obligate intracellular parasites. Virology is the study of viruses. This is a huge field. There's a lot of stuff going on there. It's a lot newer than bacteriology because we couldn't see viruses before some of the electron microscopes came up. The only viruses that can be seen with a light microscope would be some of the big pox viruses. And that's only if you're really lucky. Viral particles, also known as a virion, are extracellular forms of a virus. They exist outside the host and they facilitate transmission from one host to another. They contain nucleic acids, as we've already talked about. They're surrounded by a protein coat that's called a capsid. It's a capsid coat. In some cases, other layers of materials can be found associated with them. I think of viruses as being the traveling salesman that has to fly all around the country all the time. They learn to pack. Have you ever seen their overnight cases? They're like this. They got everything they need in that tiny little suitcase they can roll on and slap up there. They're efficient. My wife cannot pack efficiently to save her life. If we go on a 2-day trip, you can bet she's got the biggest suitcase out, and I get this much space, and the rest is hers. Well, I don't know if I'm going to need these shoes or these. What is it with women with shoes? It's like one pair of dress shoes, a pair of tennis shoes, I'm good. You know, but she's got to have sandals and flats and all that. All right, so viruses have to pack light. So what they do, have you ever gone to Lowe's? If you're married, Lowe's becomes your date night. I'm serious. I go out to eat with my wife and we wind up at Lowe's no matter what the date night's supposed to be. And I try to keep her away from the organizational aisle because she has a problem. No, I'm serious. You wouldn't understand the Tupperware things. Not Tupperware, but you know, the racks with...
You don't understand why I need those things.
Well, I understand that if I can distract her enough to keep her off that aisle, I save money. So that's what happens. All right, so you go to Lowe's, and if you look at the paving stones or the tile, sometimes you see those that they're identical, but you lock them together. They kind of snap together, so they're hexagonals, and you can snap them together and make a floor out of it. And they all look just the same, but they're just snapped to. Well, that's what I think of the capsid code. The proteins are identical, so you only need one gene for that protein, and they just snap together and make the code. It saves space. Viruses are very efficient at saving space. Okay. There's viral replication video. You could watch it on the website. I don't think it'll play here. Nope. Viral genomes, either DNA or RNA. There are some circular, but most are linear. You have viruses that can infect everything from bacteria, fungi, yeast, animal cells, and plant cells. A lot of your plant viruses are going to need a vector. A vector is some organism that can spread the disease. So plants have a hard outer coat that the virus can't penetrate unless there's damage to the plant. So if they happen to be in the salivary glands of an insect that is chewing on the plant, that gives them a way into the cell. So a lot of caterpillars and plant-eating bugs actually carry viruses in their salivary glands to attack the plants. So here's the viral genomes. You've got DNA viruses, RNA viruses, and then your retroviruses, like this one that can, it goes from RNA to DNA. It starts off as RNA, but then it can become a DNA, a double-stranded DNA in the cell, and the hapadenoviruses that are in there.
Can they go back?
Hmm?
Can they go back like from a DNA?
It does, because when it's making new copies to go out, those copies will be RNA.
So its copies can go back, but itself has to stay.
Once it's converted, many times that original RNA is chopped up in the process.
So they can go from the least stable state to the most stable.
Right. Most of
Why
your...
do they want to be more stable? Because more mutations would be...
Because your chromosome is made of DNA. So they want to insert into your chromosome and use your machinery to replicate themselves. So that's one reason they would want to do that.
But would they ever want to go to an RNA state where they could mutate and be unrecognizable and then go back?
Well, if it's part of their normal life cycle, they do that anyway. So I don't think they do it on purpose. It's just part of their life cycle. So you'll get high mutation rates in HIV. There was a crime scene investigation in Las Vegas several years ago. I haven't watched it in years. That they were able to prove a man committed murder because he committed murder. Here we go to prostitutes again. She was a prostitute, and he cut her throat, and the blood got in his eye. Well, he got HIV from it. Well, when they caught him, they tested his blood, and they looked at the specific strains of HIV, and because they were the same strains that the prostitute had, they hadn't had time to mutate yet. It was proof that her blood got onto him. Make sense? And so I don't even know how to answer your question, really.
Well, I just think about more mutations is less immune.
Yeah, but don't we all want a little stability? Sometimes a mutation can be a harmful thing. And in many of the cases, a lot of the viral particles that are mutated, there's out of 100, maybe 5% of them can infect the next generation. So you got 95 of them that are just wasted efforts on normal kind of replication cycle of the lentiviruses like HIV. I think it's about 5%. There's some animal viruses like rabies that are spread through salivary glands and through saliva and things like that. So the other animals can be vectored. Your yellow fever is vectored through bite of mosquito. Any of your biting insects can carry viruses. Ticks, fleas, etc. All right. And then they've got down here other viruses. We'll have to see what they mean by that. Viruses come in many shapes. They can be anywhere from 0.02 microns, so you don't necessarily filter those out if you're filtering medications, to 0.3 microns. That would be the big pox viruses. Viral genomes are smaller than those of eukaryotic cells. They don't have to pack everything they need. They just pack their essentials, and they use the stuff that's in the hotel room when they get there. So they don't mind using the shampoo. My wife has to carry her own shampoo. That's why I can pack so light. Capsid, that's the protein shell that surrounds the genome of the viral particle, composed of a number of protein molecules arranged in precise and highly repetitive patterns around the nucleic acid. That highly repetitive patterns is where you're snapping those pieces together. So you form kind of a shell, and sometimes the viruses look like straws that you've just wrapped that stuff around. Sometimes they look like helices. Very few of them are as complex as the bacteriophage viruses. Capsomere is a single subunit of the capsid, smallest morphological unit visible with an electron microscope. Here's one. So see, these are identical kind of shapes here, and they just snap onto the viral RNA there, and they surround it. So they're protecting the RNA, and they self-anneal into these long tube-like structures. What's that, a tobacco mosaic virus? That's the one that you use if you're teaching high school. That's one of the easiest ones to use. You take someone's cigarette, not burned, and you take some of the tobacco out of it, and you put it in water, and then you take that juice from that, and you put it on a plant that you scored with an emery board, and it will transfer the virus. Tomato plants are really susceptible. So you can demonstrate the transfer of the virus from the solution to the plant and show what effects it has. Enveloped viruses, they contain part of the outer surface. I always think of the American Indians that hunt for buffalo, and they would put the buffalo hides over them to sneak out among there. They're hiding from the immune system by wearing the skin of the host cells. So they're less likely to be caught because they're not exposing all of themselves. They're exposing very little of themselves. Enveloped viruses have a weakness, though. If you can lyse or destroy that envelope, then the virus can't replicate. So an antibody binding to that envelope and destroying it is a quick way of getting rid of those enveloped viruses. That's one thing that we try to do when we're using killed viruses. We're trying to develop antibodies against those specific surface antigens. So naked viruses don't have that membrane on the outside of them. Enveloped viruses have to have the membrane, but they also have to spike their own proteins in there. There's all sorts of symmetry associated with it. If this were a virology course, we would have labs where you had to make models of the icosahedrons, and the icosahedrons have like 120 different angles on it. Yeah, we were given, icosahedron. It gives you a symmetry that there's lots of angles to it, and that's where the nuclear material is going to be hidden inside those cells. The human papilloma viruses, the ones that cause genital warts, those are known to have the icosahedral shape. They're a big problem with turtles right now. Have I mentioned that before?
No.
Yeah, the sea turtles are getting infected. They're transmitting this STD. And the problem is turtles don't have a penis. They have a vent. So they have to get really close to transfer the material. That material can't be transferred efficiently if there are warts all around the vent area. It prevents them from getting a good connection. So it's causing a lot of infertility in those sea turtles. There are sea turtle rescue units all around the coastline of Florida and Georgia where they rescue these turtles and they remove the warts from them in order to allow them to be more efficient at breeding.
How is it not in the news?
teaching a 370 course, a writing course right now, and one of the girls in there did that last summer. She worked with the turtles, and they flipped them over and had to cut the warts off of them. But there's also some parrots that are getting STDs in South America, and it's causing infertility in certain species of parrot. Yeah, just shows that being a sexual-oriented virus is a good way of being able to transfer yourself. Okay. Well, there's the icosahedron. Now, if you'll look really close at this one right here, where it's showing a cluster of units that are in there, that's a cluster of individual folds together, and it forms something that's really kind of cool. If I'm an antibody and I'm wanting to build up a resistance to a specific receptor site. So here's a host cell. There's a nucleus. And it's got a protein on the outer surface that the virus wants to infect. The virus has to have a receptor or an anti-receptor to the receptor.
What?
The virus has to be able to recognize the receptor. If that's the door it's got to go through, it's got to be able to recognize it. So why can't we develop antibodies that block that receptor to it? If it were just another protein, we easily could. The problem is down in the middle of a valley. It's going to come to me in a minute. There's a term for this. Okay, here it is. This is the valley. That's that cluster you see up there in the bottom right-hand corner. There's a valley down in here, and X down in the bottom of the valley is where the receptor off of this cell is going to have to touch. You don't mutate the receptor site that recognizes that receptor. You mutate its canyon. You mutate the outer bits of the canyon, so it's constantly changing. This is called the canyon hypothesis. It's a hypothesis that believes that as long as you mutate this outer bit on here, you can get, the virus can keep infecting. Antibodies aren't small enough to reach down into the bottom of the valley, but the receptor on the host cell is. So you'll never be able to develop an antibody against that